Hi students, today we are going to learn the module particularly on carotenoids, and that too on carotenoids we are going to discuss more about beta-carotene, lutein and lycopene. These three are important molecules among the plant carotenoids. There were more than 600 carotenoids have been discovered so far. These carotenoids are the pigments in the plants and they have different functions in the plants also. Basically, these carotenoids, they trap the photon energy from the sun and they transfer this energy to chlorophyll molecule. In turn, the chlorophyll molecule will get excited and it will be participated in the carbohydrate metabolism. That is the main function of the carotenoids in the plant system. But for the humans, how these plant carotenoids will be helpful, we are going to learn today. The very objective of this module is to learn about the carotenoids as a plant pigments, how they are going to be useful for the human health, particularly as a vitamin A precursors as well as antioxidants to alleviate vitamin A deficiency as well as chronic degenerative diseases. These carotenoids, as I have already mentioned about, so far 600 have been discovered, but these were grouped into two categories. One as carotenes, that is beta-carotene, alpha-carotene, gamma-carotene and the lycopene. These are the carotenoids which has 40 carbon units and a polyene structure with alternate double bonds. Whereas the other carotenoids which are grouped into oxygenated carotenoids also can be termed as xanthophylls. This group has oxygen in their group. So, some of the xanthophylls are also act as vitamin A precursors. If you take the carotenoids, main interest for the human health is vitamin A. In animal foods, you have preformed vitamin A which is available, whereas in the plant foods, instead of vitamin A, you have the plant carotenoids, that is beta-carotene, which is a vitamin A precursor which is abundantly present in the plants. Once you consume this, it will get converted to vitamin A. Based on the literature, when you look at the literature epidemiologically, clinically or by animal experimentation and by the cell culture techniques, it has been found that these plant carotenoids are very useful for the human health. Therefore, this has been given a prominent picture in the human health, particularly these molecules. If you look at the plant carotenoids, basically what could be the mechanism, how they work as an antioxidant. If you think about a cell which has been continuously bombarded with the free radicals which will be generated during the metabolic process as well as with environmental effects. When there were free radicals are generated in the cell, if they are not deactivated, if they are not removed, then they will make a havoc in the cell which will oxidize the lipids as well as the proteins and more so of DNA damage which will occur, which will ultimately leads to chronic degenerative diseases. But constitutively cell has enzymes which are antioxidants like superoxide dismutase, catalase, peroxidase, glutathione peroxidase, etc. But once the free radical load is more than the constitutively present antioxidants in the cell, therefore, there is a possibility of DNA damage. Therefore, there is a need to give externally through the diet these antioxidants in the form of carotenoids, because as I have mentioned you, these are the work as antioxidants. So, when this has been proved, the other hypothesis actually mechanistically this, if you Look at the other hypothesis mechanisms of these carotenoids. They stimulate enzymes that detoxify toxins or the carcinogens, enhances DNA repair mechanisms, increases apoptosis of cancer cells, decreases cell proliferation, and possess anti-angiogenesis activity, restore gap junction communications. These are the various mechanisms have been hypothesized because of the various experimental evidence which we have seen in the literature. Therefore, such a prominent carotenoids and more so of beta-carotene as a vitamin A precursor which is abundantly present in the plant foods will be useful to tackle the vitamin A deficiency which is much prevalent as a public health significance problem. The very objective of this module is to learn about the carotenoids as a plant pigments, how they are going to be useful for the human health, particularly as a vitamin A precursors as well as antioxidants to alleviate vitamin A deficiency as well as chronic degenerative diseases. These carotenoids, as I have already mentioned about, so far 600 have been discovered, but these were grouped into two categories. One as carotenes, that is beta-carotene, alpha-carotene, gamma-carotene and the lycopene. These are the carotenoids which has 40 carbon units and a polyene structure with alternate double bonds. Whereas the other carotenoids which are grouped into oxygenated carotenoids also can be termed as xanthophylls. This group has oxygen in their group. So, some of the xanthophylls are also act as vitamin A precursors. If you take the carotenoids, main interest for the human health is vitamin A. In animal foods, you have preformed vitamin A which is available, whereas in the plant foods, instead of vitamin A, you have the plant carotenoids, that is beta-carotene, which is a vitamin A precursor which is abundantly present in the plants. Once you consume this, it will be get converted to vitamin A. Based on the literature, when you look at the literature epidemiologically, clinically or by animal experimentation and by the cell culture techniques, it has been found that these plant carotenoids are very useful for the human health. Therefore, this has been given a prominent picture in the human health, particularly these molecules. If you look at the plant carotenoids, basically what could be the mechanism how they work as an antioxidant. If you think about a cell which has been continuously bombarded with the free radicals which will be generated during the metabolic process as well as with environmental effects. When there were free radicals are generated in the cell, if they are not deactivated, if they are not removed, then they will make a havoc in the cell which will oxidize the lipids as well as the proteins and more so of DNA damage which will occur, which will ultimately leads to chronic degenerative diseases. But constitutively cell has enzymes which are antioxidants like superoxide dismutase, catalase, peroxidase, glutathione peroxidase, etc. But once the free radical load is more than the constitutively present antioxidants in the cell, therefore, there is a possibility of DNA damage. Therefore, there is a need to give externally through the diet these antioxidants in the form of carotenoids, because as I have mentioned you, these are the work as antioxidants. So, when this has been proved, look at the other hypothesis mechanisms of these carotenoids. They stimulate enzymes that detoxify toxins or the carcinogens, enhances DNA repair mechanisms, increases apoptosis of cancer cells, decreases cell proliferation and possess anti-angiogenesis activity, restore gap junction communications. These are the various mechanisms have been hypothesized because of the various experimental evidence which we have seen in the literature. Therefore, such a prominent carotenoids and more so of beta-carotene as a vitamin A precursor, which is abundantly present in the plant foods, will be useful to tackle the vitamin A deficiency, which is much prevalent as a public health significance problem. As we have seen that carotenoids, particularly the beta-carotene, as a vitamin A precursor, which is much useful, particularly for the vegetarian people, which is the primary source of vitamin A. But the absorption and bioavailability is really interested to know about these vitamin A precursors and particularly beta-carotene, which we are talking. Bioavailability is the fraction of an ingested nutrient that is available for utilization in normal physiological functions, metabolism and or the storage. In the context of this discussion, bioavailability of carotenoids is defined as the amount of these micronutrients that are absorbed by the intestinal enterocytes and transported in the bloodstream. Once this is get absorbed, the beta-carotene will be converted to vitamin A. Vitamin A means that is retinol, both alcohol form and the aldehyde form, which are interconvertible, will be formed in the presence of 15 prime monooxygenase and other reductases. Theoretically, beta-carotene, when it is cleaved centrally, it will produce 2 molecules of vitamin A. But practically, because of the absorption and other problems, beta-carotene practically cannot give 2 molecules of vitamin A. But we should also understand the other factors which will affect the beta-carotene retention in the plant foods. Particularly, we have a habit at household level that when we purchase the green leafy vegetables, for example, we cut the vegetables and then wash the vegetables. This is normally the practice which at household level which we could observe, but that should not be the correct practice. One should really first wash the vegetables, then the vegetables should be cut into smaller to the medium pieces, then only it should be cooked by different processes. Usually people cook into a smallest possible pieces where the area of exposure to the oxygen will be more, therefore the oxidation to the beta-carotene occurs, therefore the less retention of beta-carotene in green leafy vegetables, for example, will be lower before it is further cooked or processed for consumption. So, various cooking methods also are available in our country. The people, the low socioeconomic group people, they will cook these vegetables in the vessel even without keeping the lid, and some people will keep the lid. Also steam cooking or the pressure cooking, sauteing with little oil or the deep frying, and also dehydration either by a direct sun drying or by a shade drying or of the modern ways of solar cabinet drying are also available. Also microwave cooking, a modern method of cooking, is also nowadays available with upper middle class and the higher income group people. But in the literature, it has been shown very clearly cooking the green leafy vegetables with lid in a way better in a steaming or on par with steaming. Microwave cooking also, green leafy vegetables retained the highest possible beta-carotene, even though there were losses about 30% with various cooking methods to this. As we have discussed about the bioavailability, once the beta-carotene is ingested through the diet, the beta-carotene will be absorbed in the small intestine. Through the enterocytes, it will be circulated and it will get converted in the presence of enzymes to vitamin A. But because of the matrix and the host other factors, this beta-carotene is unable to give 2 molecules of vitamin A. In Indian conditions, it has been shown that 8 molecules of beta-carotene are providing only one vitamin A molecule. Therefore, the current RDAs, for example, of 600 micrograms of vitamin A, which is equivalent to 4,800 micrograms of beta-carotene. Therefore, one should advise the people that they should consume 4,800 micrograms of beta-carotene every day to get their vitamin A levels. If you look at the world picture about the vegetarian and non-vegetarian consumption, and particularly our Asia, more so of our country, predominantly we are vegetarian people. Our population, more than 85%, consume plant foods for our energy and other purposes. Therefore, primarily our vitamin A requirements comes through the plant foods only. Therefore, assessing the plant foods for their vitamin A or beta-carotene levels is important. In this direction, as a student, what we have to learn is the beta-carotene rich foods which we have to look at those, and those foods only we should consume. Also, we should advocate for the vitamin A levels. If you look at the list of green leafy vegetables available in this country, there were many green leafy vegetables commonly consumed, but very commonly consumed, if you could list it, approximately 20 to 30 green leafy vegetables are available across the country. Among them, drumstick leaves, the richest source of beta-carotene. Also amaranth, spinach, hibiscus, curry leaves, mint leaves. These are the other green leafy vegetables are also rich sources for the beta-carotene. For example, if you take from 10 grams to 100 grams of any vegetable for that matter, which will give the vitamin A requirements for the various groups of our Indian community. So, the other rich sources of the beta-carotene in the vegetable groups, what we have discussed earlier about the green leafy vegetables, now I am mentioning about the vegetables like tomato, pumpkin, green chilies as well as carrot are also rich sources for this beta-carotene. For example, if you take in the tubers list, the carrot is also very rich source. It has more than 6,000 micrograms of beta-carotene. Even a one carrot, or sometimes if the carrot is bigger, a half carrot can give you the vitamin A requirements of children of this country. The fruits is also rich sources of this beta-carotene molecule, particularly the papaya, mango, tomato. These are the fruits which are abundantly present in this beta-carotene. For example, if you take the papaya, which has 880 micrograms of beta-carotene, papaya has also another xanthophyll called beta-cryptoxanthin, which is also a vitamin precursor, which is abundantly present, this particular carotenoid in this fruit category. Therefore, increasing the consumption of foods rich in certain carotenoids, as we have discussed, like beta-carotene and other things, in particularly dark green leafy vegetables, may decrease the risk of developing advanced or exudative age-related macular or degenerative disease, that most widely and visually disabling form of macular degeneration among aging population, that is older population. As we have discussed about the vitamin A precursors, that is beta-carotene, which we have a problem of vitamin A deficiency, particularly in the children, and we have the problem of age-related macular degenerative disease during the aging. Suppose the people who are living more than 70 years of age, those have this problem of AMD is going to occur. The macular region of the retina, particularly there are two pigments are concentrated. One among them is the lutein. Lutein is a xanthophyll. Along with the lutein, there is another molecule, zeaxanthin is also another xanthophyll carotenoid, which is present in the macular region of the eye. Actually, this lutein and zeaxanthin, these two compounds will filter the UVB radiation and they act like a goggles. Actually, they prevent the UVB oxidation of the retina. Therefore, they are actually macular degeneration has been inhibited because of these two pigments are concentrated in that area. Therefore, What we have to advocate is, for vitamin A deficiency, you should think about the beta-carotene for the children and other age groups, whereas in the aged population, age-related macular degeneration can be prevented, can be postponed by feeding with lutein and zeaxanthin rich sources of green leafy vegetables, fruits and roots. As I have mentioned about the lutein, if you know little bit about the lutein, these are the xanthophyll carotenoids which are abundantly present in the dark green leafy vegetables. As I have told you about the beta-carotene which are abundantly present in the dark green leafy vegetables, on par with beta-carotene or more in concentrations in green leafy vegetables, lutein is abundantly present. Therefore, once you are choosing for the beta-carotene, you are also choosing for lutein and zeaxanthin also from these sources. Another important source for the lutein is about the egg yolk. Egg, though it's a animal food, which is also concentrated not only the beta-carotene but also the lutein, which is highly concentrated in the egg yolks. Therefore, by consuming eggs, you have a double advantage of both vitamin A, beta-carotene as well as lutein can be met through eating the eggs. If you look at the rich sources of the lutein, drumsticks, curry leaves, radish leaves, betel leaves, coriander, mint and spinach and colocasia, these are the leaves where luteins are in high levels. Another molecule which we are going to discuss about the carotenoids is the lycopene. Lycopene, if you could observe, if you think about lycopene, the first thing you should get into your mind is about the tomato. The tomato red color is because of the lycopene. So, this lycopene is not a vitamin A precursor, but it is a powerful antioxidant. This lycopene is also belongs to the carotenes group of the carotenoids, and it is in the basically in the plant system, as I have mentioned, it traps the photon energy and also in the photosynthesis protecting the plants against photosensitization. So, these compound, particularly the lycopene, if you compare with the beta-carotene, this molecule has open-ended rings where the double bonds are exposed. Therefore, oxidation takes much easier through this molecule than of any other plant carotenoids. Therefore, it has been proved lycopene is a powerful antioxidant. Lycopene, when we have looked at the lycopene from the various sources also, it has not only the all trans position, that means the compound will be without any kinks at the double bonds, whereas some of the cis carotenoids like cis isomers of this lycopene are also present in the tomato and other sources. And in the literature, it has been shown that some of the cis carotenoids are powerful antioxidants, particularly against cancer. So, lycopene is the most efficient singlet oxygen quencher and phenoxyl radical scavenger among the naturally occurring carotenoids. If you list the all carotenoids from this list, if you could see it, lycopene stands number 1 as a singlet oxygen quencher, which actually deactivates the free radicals so that it will attack the free radicals very efficiently comparatively other this thing. So, if you look at the geometrical isomers of lycopene, the all trans lycopene, 5-cis lycopene, 9-cis lycopene, 13-cis lycopene, 7,9 7' cis lycopene, like this there are many cis isomers are also present naturally, as well as this will be formed during the cooking and processing methods also. And lycopene is more bioavailable from tomato paste than from fresh tomatoes. So, very important thing what we have to learn about the carotenoids, particularly when you are looking at the lycopene, the processed product, the tomato uncooked or the raw one which has a lycopene, and if you consume that, or the tomato which has been cooked, paste or sauce which has been made, from that source lycopene is more bioavailable, probably because of the matrix associated problems has been removed while processing these tomatoes. So, the mechanism of action, if you could take, as I have mentioned about, this will, because of this, it will, reactive oxygen species will be reduced and the antioxidant potential is very high, oxidative damage will be lowered and the oxidative stress to the cell also will be lowered. Therefore, the chronic degenerative diseases like cancer risk, cardiovascular disease risk will be reduced because of the actions of lycopene. Far from being inert, by virtue of its unique chemical properties, studies have shown lycopene to possess superior antioxidant abilities in comparison to other carotenoids. It has the ability to quench singlet oxygen and prevent oxidative damage to other molecules and cellular structures because of its unique structure of 11 conjugated double bonds and no cyclic groups. Because of this role, many researchers now believe that lycopene may profoundly influence the evolution of several chronic diseases in tissue-specific manner. The sites of concentrations of this lycopene in the humans, if you could look at various organs like adrenal glands, testes, liver, prostate gland, this molecule will be concentrated. Because of this lipophilic nature, these molecules, lycopene tends to accumulate in various tissues. So, predominantly, as I have mentioned, prostate, liver, adrenal glands and testes, with lycopene accounting for up to 80% of the total carotenoids in these latter two tissues. Lycopene concentration in the testes and adrenals is up to 9 times higher than in the tissues, with the second highest amount will be in the liver. So, you might have heard about the prostate cancers. Strongest known link between lycopene and the chronic disease have been mentioned. From the health professional follow-up study conducted from 1986 through January 31st, 1992, it was suggested that frequent intake of tomato products or lycopene is associated with reduced risk of prostate cancer. Therefore, one should advocate to eat the tomatoes. But when you look at in the market, you will find the varieties of tomatoes. Tomatoes of different color also. Tomatoes with the green, tomatoes with yellow, tomatoes with the bright red, tomatoes with the dark red. So, these are the shades. So, one should really look into the lycopene concentrations of the tomato. But usually, if you could look at the brightly red color tomatoes are the rich sources for the lycopene. For example, the other foods, not only the tomatoes contains approximately 3.1 to 7.7 milligrams per 100 grams, whereas guava fruit is around 5.4, grapefruit 3.36, watermelon 4.1, apricots dry 1.2 milligrams, and papaya fresh, which has red color inside, also contains lycopene. In addition to beta-cryptoxanthin and beta-carotene, which I was mentioning, papaya also contains lycopene between 2 to 5.3 milligrams per 100 grams of the food. In conclusion, so far what we have discussed about the plant carotenoids. So far 600 carotenoids have been discovered. These were made into two categories like carotenes and oxygenated carotenoids, that is xanthophylls. In India is facing the problem of vitamin A deficiency. WHO recognized as public health significance problem because of the 0.5% beta spots prevalence is still continuing in India. To alleviate vitamin A deficiency, one should choose either vitamin A or the precursors of vitamin A which are abundantly present in the plant foods. Therefore, beta-carotene, which is a vitamin A precursor, is present in the dark green leafy vegetables, fruits, roots and other vegetables also. If you look at the rich sources, drumstick leaves, amaranth, spinach, from the fruits list, papaya are the rich sources for beta-carotene. 4,800 micrograms of beta-carotene will get converted to 600 micrograms of vitamin A, which is the recommended dietary allowances for Indians for vitamin A. We discussed other molecule, the lutein. Lutein and the zeaxanthin, another xanthophyll, which is concentrated in the macula region of the retina of the eye. These two pigments actually works as goggles which prevents the UVB radiation from the sun. So, therefore, there is no oxidation to the retina, therefore, there is no damage to the macula, therefore, there is no age related macular degeneration in the aged population. Therefore, we have to choose rich foods of lutein from the foods list. Particularly, if you could choose for the beta-carotene rich foods, you are also choosing for the lutein rich foods. Both these drumstick leaves, amaranth, spinach, coriander, mint, all other colocasia leaves are rich sources of lutein. Another molecule which we have discussed is about the lycopene. The word lycopene should recollect you about the tomato. Tomato is abundantly present, this lycopene, which is more bioavailable form is the processed product. Therefore, consuming the more lycopene rich foods will prevent the prostate cancer particularly. Therefore, consuming green leafy vegetables, roots and tubers, which are rich sources for the plant carotenoids, which has been shown as antioxidants. They not only prevent the vitamin A deficiency, also the age related macular degenerative disease and other chronic degenerative diseases in the humans. Thank you.